Okay. Hello, everybody. Welcome to another edition of PaleoPerks. Today, we will be joined by Melissa Kemp from the University of Texas in Austin, who will be giving a talk on the extinction, colonization, and conservation in a biodiversity hotspot, lessons from the Caribbean fossil. Before we get started, just a quick few announcements. Talking about the format of today's seminar, we will have a few minutes dedicated to welcomes and announcements, followed by Melissa's talk, which will be around 30 minutes. Following that, we will have a moderated question and answers for around 10 minutes. And don't forget to send your questions during the talk via chat to the Questions PaleoPerks host, who today is Elizabeth. A few housekeeping rules before we get started. PaleoPerks values the participation of all folks interested in paleosciences. If you have not gone through our code of conduct, please make sure that you have. You should have. If you are in this viewing room, you should have already gone through that. You should be muted during the duration of the talk. If you find yourself for some reason to be unmuted, please mute yourself so Melissa can give a dedicated talk. And as always, any questions that you have, feel free to send them to the questions at PaleoPerks host, or you can also use the raise hand function, and then we will unmute you and you can ask your question directly to Melissa. We also have closed captions built into Zoom. You can use the CC button to show or hide them. If you know any early career researcher, please nominate them. The link for that should be dropped into the Zoom chat momentarily. And there will be another link that we collect weekly feedback form for demographic information. It is completely anonymous, but optional and highly encouraged. You can also find that in the chat window. And with that, a little bit about today's speaker. We have Melissa, who did her bachelor's in biology from Williams College, followed by a PhD in biology from Stanford University, following which Melissa did was an environmental postdoctoral fellow at Harvard University, and since September 2018 has been an assistant professor in integrative biology at the University of Texas in Austin, where she is also the director of the Inclusive Student Training in Collections and Field-Based Topics Summer Undergraduate Research Program. And with that, Melissa will talk to us about Caribbean vertebrates and their extinctions and what we can learn about them to help shape conservation policy. And with that, Melissa, please feel free to take over the screen share.
Great. Thank you for that introduction, Natasha, and good day, everyone. Thank you for coming to my talk today. Okay. And I believe everyone should be able to see my screen now. Is that correct?
Yes.
Okay, fantastic. So today I'm going to be talking about extinction, colonization, and conservation in a biodiversity hotspot, lessons from the Caribbean fossil record. And before I get started, I want to make sure we're all on the same page as to what biodiversity is. So very briefly, biodiversity is a variation in life on the planet. So we often think about it as organisms that are found in an ecosystem at a given time point. But it's also the functions that organisms possess in their environments. And I'll talk a little bit more about that at the end of my talk. Biodiversity loss is one of the biggest challenges facing humanity today. The loss of species and their ecological functions threaten the ecosystem services that we depend upon, services like food production, pest control, pharmaceutical development, and nutrient cycling, to name a few. But biodiversity also plays a really important role culturally, spiritually, and also in recreational practices that human societies participate in. So it promotes not only physical and mental well-being, but also emotional well-being. So there is a really urgent need to characterize the existing biodiversity that we have on the planet, quantify recent biodiversity loss, and determine how species and ecosystems will be impacted by ongoing and future global change. And in terms of my research interests, I study how vertebrates respond to global change phenomena like the ones in this figure. So habitat loss, invasive species, climate change are primary foci of my research program. And I'm interested in the interplay of extinction, colonization, and the persistence and diversification of species through time and space. I work primarily with lizards in the Caribbean, although I have started to work on lizards in Texas. But today I'm going to be talking solely about my Caribbean research program. And that's because the Caribbean is a really unique region. It's a biodiversity hotspot. And like many other insular systems, biodiversity hotspots make up a very small percentage of the Earth's surface. So currently there are 36 biodiversity hotspots that have been recognized, and together, they're about 2.5% of the Earth's surface, but they contain more than half of all plant species and over 40% of all terrestrial vertebrate species. And in many cases, those species are endemic, meaning that they're found nowhere else in the world. And the Caribbean is a really great example of a biodiversity hotspot, particularly with regards to its lizard fauna. The ecology and evolution of many of the extant Caribbean lizards have been well studied by scientists, but much less is known about the ancient lizard community, even though there is historic and paleontological evidence that extinction and colonization have taken place in this archipelago. So my research integrates paleontology and zooarchaeology with observations of modern-day ecosystems to figure out what the fossil record tells us about ancient biodiversity and how present biodiversity has been shaped through time. And for the rest of my talk, I'm going to present vignettes of my work on how biodiversity is shaped by extinction and colonization. And these processes are not acting in isolation. I'm also interested in the interaction of extinction and colonization to shape the present day biotas that we have in the Caribbean. But first, I want to spend some time talking about the Caribbean fossil record, because the Caribbean isn't the first place that one typically thinks of when they think of paleontology. The Caribbean is a geologically complex region composed of the Greater Antilles, the Lesser Antilles, which is this volcanic island arc east of Puerto Rico, and the Bahamas, which are north of Cuba and Hispaniola. So because of the heat and humidity of this region, fossilization is pretty poor. We lack a lot of deep time fossil records for the region. Lizards, for example, are only known from a few Miocene-aged amber specimens from the island of Hispaniola, which is where Haiti and the Dominican Republic are. These species have played an integral role in our understanding of diversification rates and biogeography for this group, the Anolis lizards. But because these fossils are spatially restricted and rare, they have limited utility compared to the Quaternary age fossils of the Caribbean, which is what I study. So while encompassing only the most recent slither of geological time, the Quaternary fossil record is much more widespread with both greater spatial and temporal and taxonomic coverage. So every island in the Greater Antilles has Quaternary deposits, as do many islands in the Lesser Antilles and several islands in the Bahamas as well. And most of these deposits are cave deposits, although there are some really compelling sinkhole underwater deposits as well. And the stratigraphy that you find, particularly in these cave deposits, allows us to reconstruct faunal assemblages through time. And what's especially great about the Quaternary fossil record is that we generally know when extinction and colonization drivers were shaping these communities. So there was significant climate change during the Pleistocene-Holocene transition about 12,000 years ago, and sea level rose to today's current levels, meaning that in the past, island area was not only greater, but land masses were also closer to one another. And we can see that in this figure here, where this light mint color represents the extent of land masses at the last glacial maximum. And then this darker green color represents current island area. So with land masses being larger in the past, species richness may have been higher due to species area relationships. Colonization rates may have also been higher in the past due to the increased connectivity of terrestrial habitats and the proximity of places to one another. These sites also document the arrival of humans into the Caribbean during the Holocene, as well as the ways in which they modified natural habitats and the species that they introduced. And the Caribbean was colonized in multiple ways by a variety of different cultural groups. So there's a real interesting cultural heterogeneity that allows us to test hypotheses as to how different human practices might affect biodiversity and what others might consider a replicate system, with these islands being similar in size across the archipelago. So all of this information is critically important, though, because this is the only time in geologic history where the main drivers of biotic change are humans and climate. And thus the Pleistocene and the Holocene provide insight into what we might expect during our current epoch, the Anthropocene. So in the Anthropocene, we expect to see more habitat modification increased species introductions, global warming that will continue to change sea level, more intense storm activity, and humans are now the dominant influence on climate and the environment. All of these factors will impact today's biodiversity, but the fossil record gives us insight into how these factors have impacted biodiversity in the past. And what's especially exciting about Quaternary fossil records compared to deeper time fossil records is that many of the taxa that we describe from Quaternary deposits are extant ones. So we can reconstruct perturbation response trajectories for species and individual populations using data from before, during, and after an environment changed. And we also have contemporary data from extinct taxa, so we can compare and contrast how different taxa are responding to the same threats, which might be instructive for understanding future responses to similar threats. So first, I'm going to talk about some of the work I've done on body size, and body size has been foundational to my research program. I'm really interested in the relationship between extinction and body size because body size plays a really important role in a lot of life history traits of a species. So where it can live, what its reproductive output will be, what its metabolism will be. And it's also something that is recorded in the fossil record or easily estimated from fossil materials, unlike some of the other life history traits I might be interested in studying. So there have been studies in a host of taxonomic groups indicating that extinction has been size biased during the Quaternary, with a tendency for large-bodied species to go extinct. But prior to my work in the Caribbean, very little had been known about the relationship between body size and extinction in lizards. Prior to this work, researchers used metrics like the IUCN list to get a sense of extinction, but not all species have been evaluated by the IUCN, and there is an overrepresentation of large charismatic lizards in that database. But if we use the fossil record, we have a true dichotomy between what is extinct and what is extant, and then we can articulate the actual extinction trends that exist. So that's what I've endeavored to do with my work. I created a database for all Caribbean lizards, past and present, and it includes snout vent length, a commonly used body size metric shown here with this red arrow. All of these data were taken from the literature. And in the case of the extinct species, body size estimates are often reported in the literature, but these fossils are disarticulated. So estimates are typically derived from body size estimation equations that are made on comparative material of extant related taxa, where you might measure an osteological character, such as the length of the dental row on this dentary. And we know how that length changes relative to snout vent length in extant taxa. So we can make an equation to estimate snout vent length in our fossils based on the measurement of the dental row. So I noted whether each species was extinct or extant. And I also noted the family of each species because shared life history traits of closely related species might predispose some lineages to extinction. And in this database, I had about 416 species, representing a little less than 5% of all lizard species worldwide that had been described at the time. And in this database, 13 of those species are extinct. And when I compared the body sizes, I found that the extinct ones, which are shown here in this red distribution, are significantly larger than the extant ones shown in white. However, extinction is not evenly distributed across the phylogeny. In fact, there's a significantly higher proportion of extinctions in the family Leiocephalidae, the curly-tailed lizards, than expected. And in fact, this family has been completely extirpated from the Lesser Antilles, and I'll talk about that a bit more in a moment. And if we look at body size within Leiocephalidae, though, we do see that the extinct lizards are significantly larger than the extant ones. And there are 4 other families in the Caribbean that have confirmed extinctions. And in all but 3 cases, the extinct species are among the largest, though the difference is not significant. The one exception being for the iguanidae. These are some of the largest lizards in the Caribbean. All but the green iguana, which is introduced, is endangered. And one of the largest lizards in this family, Iguana delicatissima, is highly extirpated throughout its historic range. So there certainly is a decline in this population at the very least. It's also important to mention that many of the species in these families are understudied, and additional data from extant populations need to be collected so we can continue to address what appears to be a non-random nature of extinctions that the fossil record helps us to uncover. So this work represents a first pass at understanding extinction trends, and there are a lot of really interesting directions in which we can go. One thing that we would benefit from is more detailed extinction chronologies, which help us narrow in on exactly when a taxon went extinct and what may have caused its extinction. And this has been a major focus of research in the non-volant Caribbean mammal fauna of the Caribbean, which was quite large prior to the Holocene. Scientists have meticulously radiocarbon dated fossil specimens to get last occurrence date estimates. And when you combine this data with biocide data and archaeological data, some interesting trends emerge. First, we find that there are two distinct waves of extinction. The first wave of extinction coincided with the arrival of indigenous groups in the Caribbean, and that's when you lose large-bodied taxa like the endemic primates of the Caribbean and the large sloths. The second wave of extinction occurs during European colonization, and at that time you lose small-bodied taxa like rice rats and Nesophantes, which is a shrew-like insectivore. But unfortunately, we don't have this type of temporal resolution for Caribbean lizards. They haven't been radiocarbon dated as extensively. But such data sets would help us disentangle the role of climate change and human colonization in the extinction events that we see. That said, I do want to point out that for many of these extinct taxa, we have museum specimens. So that seems to suggest that at least some of these extinctions are happening after European colonization, when people were starting to collect specimens for museum collections. We can also start to investigate the ramifications of these extinctions on other aspects of the ecosystem, which is something that my lab has been doing with regards to community structure, not only in the Caribbean, but also in Texas. And I'm going to run through an example of this type of work in the early Holocene fossil record of Anguilla, which is a small island in the Lesser Antilles, so right west or east of Puerto Rico. It harbors this really interesting early Holocene site called Catouche Bay. And during the late Pleistocene, when sea level was lower, Anguilla was connected to St. Martin and St. Bart's as part of a much larger island. And it's suggested that during the Pleistocene Holocene transition, 98% of this land mass was lost due to sea level rise. So when you see a dramatic loss in island areas, such as what Anguilla and its neighboring islands experienced, you might see extinction occurring from island area loss alone because of the species area relationship. So this site had been excavated previously, and researchers previously had only identified two lizard genera at the site, Anolis and Leiocephalus. But I revisited some material and I identified fossils from two other genera, Phyllidiscalcis and Thecodactylus. So Thecodactylus, Phyllidiscalcis, and Anolis are still found on Anguilla today, but Leiocephalus is extinct. As I mentioned earlier, it's extinct throughout the Lesser Antilles. So this is now a great system with which we can assess how the removal of a species, particularly a large-bodied species, affects the rest of the community. So I calculated the evenness of the Catouche Bay community through time using a metric called the Pylos Evenness Index. Evenness refers to how close in proportion the number of species in a community are. An even community with 4 species in equal proportion looks like this and will have an evenness index of 1. When evenness decreases, one or a few species begin to dominate, and the metric will also decrease. So let's look at the evenness of the Catouche Bay assemblage through time. And I've plotted the last occurrence of Leiocephalus on the graph in red, and that occurs about 6,000 years before present. And one of the first things that we see is a drastic drop in evenness, a 53% drop in evenness between 9,000 and 8,000 years before present. And when you see a decline in evenness so drastic like that, it usually signifies that the community has lost some stability. And indeed, not so too long later, we see the last occurrence of Leiocephalus. And after the last occurrence of Leiocephalus, Anolis becomes the most dominant taxa, consistently accounting for 80 to 90% of the population, while Phyllidiscalcis and Thecodactylus persist, but in much lower abundances. So I was really interested in this dynamic we were seeing with Anolis and Leiocephalus, and by looking at the ecological literature, it's made me start to wonder if the loss of Leiocephalus was a predator release for Anolis. And I made reference to the modern ecological literature because if we look at it, we do know that Leiocephalus eats anoles in areas where they co-occur today, as this photo illustrates. But there's been also a lot of empirical studies looking at the relationships between these two organisms, particularly in the Bahamas. And empirical data from those ecological studies confirm that Anolis is negatively impacted by Leiocephalus where they co-occur. So when Leiocephalus is introduced to islands with anoles, which is represented by the white circles, anole density decreases relative to islands without Leiocephalus, which are these open circles. We also know that Leiocephalus causes anoles to perch higher in vegetation, presumably to reduce interactions with this terrestrial predator. So an implication of this paleontological data, together with modern day ecological data, is that the high anoles or high density that anoles reach today, especially in the Lesser Antilles where Leiocephalus has been completely extirpated, may be the result of a decrease in predation in this system. So now I want to switch gears and talk a bit about colonization. And I'm going to specifically talk about some work that I've done with several colleagues, former undergraduate student of mine, Jetta Wadman, as well as Dr. Alexis Mikhailo and Dr. Amy Goldberg, both of whom are assistant professors at Middlebury and Duke, respectively. And together, the four of us synthesized the archaeological and paleontological literature from the Caribbean to create a database of vertebrate species introductions. And we focused on terrestrial vertebrates. And we delved into the ramifications of these introductions on present-day Caribbean biodiversity. And this particular figure highlights only the taxa for which we have a first introduction date. And unsurprisingly, we can see that the number of introductions increases towards the present in every single taxonomic group that we examined. We define 3 time periods of interest in the study: the indigenous time period that's prior to European colonization. We have a colonial time period, and then we also have a modern time period. And the modern and colonial time periods are delineated by the Industrial Revolution, so 1800, because that's when we start to see some significant economic and demographic changes in the Caribbean. So we can look at these data more deeply and ask about the geographic origin of these taxa and why they're being introduced. And when we do that, some patterns begin to emerge. So in this figure, each line represents a species that was introduced to the Caribbean, and they're colored based on the time at which it was introduced. The top of each of these panels represent the biogeographic realm from which a species originated. So the Neotropics, Nearctic, Palearctic as examples. And the bottom represents where in the Caribbean it was introduced to. So we have the Bahamas, the Greater Antilles, and the Lesser Antilles. And here in gray, I've plotted the taxa for which we do not have an introduction date. As you can see, this is not an insignificant number, although it does vary by group. We can then overlay introductions that occurred during the colonization of the Caribbean by indigenous people. And as you can see, mammals dominate these introductions. These organisms include things like the hutia, which is a rodent related to the guinea pig. And most of these animals were likely introduced as food sources. There is one line in the yellow, for the line that's yellow in the reptiles, and that's for the iguana, which was also used as a food source. And all of these animals are being introduced from the Americas. We can see a continued increase in introductions during the colonial period. We start to see more taxonomic breadth at this period as well. Species are being introduced not only for food, but also because there is an explicit desire to design new ecosystems, as exemplified by naturalists like Felix Herminier, who collected fauna from Puerto Rico and West Africa and intentionally introduced them to islands like Guadeloupe in some early acclimatization studies to see if those animals could survive in this new environment. They were also introduced for the Caribbean to start feeling more like home to the Europeans who are now occupying it. So they brought in animals like goats to hunt, songbirds, and things like that. And when we get to the modern era, we see that introduced species are coming from every corner of the world, which contrasts very sharply with indigenous introductions, where introduced species are exclusively from the Nearctic and the Neotropics. The increase in reptiles and amphibians is mostly attributed to the exotic pet trade and the nursery industry. And there's also a lot of intra-Caribbean introductions due to the shipping trade that now is operating between islands. So what are the ramifications of these species introductions? We go through a lot of case studies in the paper, but I'm going to provide a very broad overview here. And one of the most critical impacts of species introductions is the extinction of native fauna. A great example of this with regards to reptiles is the introduction of the mongoose. The mongoose was introduced to the Caribbean during the colonial period as a pest control agent on sugarcane plantations, and it was actually introduced to help eradicate another introduced species, the black rat. And it's a prime example of how introductions compound on one another. It was not effective at controlling the rat population, but instead wreaked havoc on native bird and reptile populations. And if you look at the timing of the introduction of mongooses throughout various islands in the Caribbean, you can see that in many places, skinks, one of the terrestrial lizards found in the Caribbean, start to disappear shortly after mongoose introductions. We also see the homogenization of biotas. So extinction combined with colonization, as I'll talk a bit more about in the next few slides, leads to the loss of native taxa, and we get more cosmopolitan taxa in these areas where they are no longer found. And indeed, when we get to the modern era, we have organisms from all different biogeographical realms of the world occupying the Caribbean. But introduced species don't necessarily have to be bad for the ecosystems. And that's why I refer to these species as introduced as opposed to invasive, which has negative connotations to it. There has been some exciting and interesting questions about the restoration of ecosystem services and how introduced species might provide ecosystem services that may have been lost due to extinction. So as I mentioned earlier, the Caribbean lost much of its mammalian fauna during the Holocene, and mammals like sloths, hutias, and bats provide really important ecosystem services like seed dispersal and pollination. The introduction of new mammals and birds and possibly even reptiles, though, may restore some of the ecosystem services that have been lost. But this is something that really needs further investigation. And this is something that I've become really interested in with my research. And I've started to query the functional diversity of reptile communities using extinction and colonization data. So functional diversity is a range of functional traits within a community. Functional diversity can be informative of ecosystem services that species provide to the environment. And reptiles in particular are very important predators. They're insectivores that contribute to pest control. They also play an important role in seed dispersal and pollination. I think people are just beginning to recognize how important of a role that is. So I sought to quantify functional diversity in reptile communities and ask, to what extent has functional diversity been lost through extinction and gained through colonization? I focused on several islands throughout the Caribbean that have paleontological data on the Quaternary reptiles. Several of these islands sit on the same island bank. So during the Pleistocene, they're part of larger continuous land masses and therefore serve as good replicates. And in this particular data set, Antigua and Barbuda are on the same island bank and serve as good replicates. And then we also have La Desirade and Grande Terre on the same island bank. So for each of these islands, I created presence-absence matrices for 3 time points. We have an ancient community, which combines extinct species with the extant native community. There's an extant native community, and we also have a modern community, which combines the extant native community with introduced species. And there are really invaluable resources building off of the years of natural history work that researchers have done on reptile communities in the Caribbean. There is a really great list of fauna for the different islands in the Caribbean. I've supplemented that with recent paleontological literature. We also have really great trait data coming from a variety of sources, most notably this book, Natural History of West Indian Reptiles and Amphibians. And I compiled trait data particularly focusing on diet, habitat affinity, foraging mode, reproductive mode, activity time, and body mass. And because the Caribbean has really high species richness in terms of reptiles, and many species share trait combinations, I group the species into functional entities. Functional entities are groupings of species with the same trait value. So a herbivorous terrestrial, 2 herbivorous terrestrial viviparous diurnal species that are large would be grouped in the same functional entity, as an example. So all of these analyses are conducted on those functional entities, taking into account where in which they lived in the Caribbean at any given time point. And what I found when I grouped species as into functional entities is that many species are FE singletons, meaning that there's only one species in a functional entity. And this histogram shows the distribution of species in functional entities. And these are all the functional entity singletons. Functional entity singletons are particularly vulnerable to extinction because they lack redundancy or other species with the same functional trait profile. So when you lose a functional entity singleton, you're losing that functional entity completely. What I also found is that the number of functional entities, as well as functional redundancy of functional entities, is higher on larger islands. In this case, islands that are greater than 2,000 kilometers squared. And what I see when I start thinking about functional richness is that there's a significant loss in native functional richness on small islands, meaning that we've lost native functional entities in these places. However, because there's no difference in functional richness between the ancient and modern islands, the ancient and small modern islands, it appears that functional richness levels have been restored by some of the species introductions that have taken place, which is really interesting to think about with regards to functional restoration of functional services. So we can look at each island individually and see how native functional diversity is lost by looking at the percent of remaining native functional entities. And when we do that, we see that large islands that have higher functional entity redundancy appear to be most resilient. Redundancy, in fact, is serving as a buffer to functional diversity loss. So if you lose one species in a functional entity on a large island, there's likely more in that functional entity. So you're not losing that functional entity completely, but that's not happening on many of the small islands. However, human land use does play a role in shaping present-day functional diversity. And we see that on several of the small islands, such as Sombrero, Mona, Culebra, and Middle Caicos. All of those retain about 80% of their native functional entities. And one thing that's really important about these islands is that they are somewhat unique in terms of their human demographic history. All of them are either sparsely populated today or are unpopulated, and they've had very small populations through time. And in many cases, those populations have been sex and age biased, where you didn't have women, you didn't have children on those islands because the only type of humans on those islands were men who were involved in activities like piracy, mining, fishing, and military exercises. So you don't have a history of large-scale agricultural development and deforestation on those islands. You don't have long-term settlements and sustenance activities that might have led to habitat destruction and the extinction of native fauna. And on those islands, you don't have introduced, some of the introduced predators like the mongoose, because you don't have those activities like agricultural development occurring on them. So I also wanted to evaluate the distribution of introduced and extinct species in functional trait space. So we talked about how richness is not really different between introduced or the modern assemblages and the ancient ones. But what if the introduced species are occupying a different area of functional trait space than the extinct ones? That might indicate that the functions are different from one another. And when I do that, I find that the introduced functional entities do indeed occupy a different part of functional trait space than the extinct functional entities occupy. And it's especially present on this principal coordinate axis 3, where you see part of that functional trait space occupied by the introduced species is an area that the extinct species do not occupy. There is some overlap with some of the introduced species, particularly the iguanas. They overlap in functional trait space with some of the extinct iguanas, but there are other areas of functional trait space like that occupied by the giant tortoises that used to live in the Caribbean, some of the largest reptile herbivores of that system. And those areas are no longer occupied by native fauna or introduced fauna. So these results suggest that functional redundancy has shielded ecosystems from significant functional diversity loss in the past. However, functional redundancy is being eroded. And when we get new species in the system, they're not replenishing those native functional entities. So many native functional entities in the communities that they support are vulnerable to extinction and functional collapse because of this shift in functional trait space. So to conclude, what my research has shown is that quaternary lizards and their extinction events are non-random and size biased with a tendency for large body species to go extinct. And these extinction events lead to significant shifts in community structure. Species introductions are a long-term phenomenon in the Caribbean, not something that's just happened in the modern era, but it's been going on since the arrival of indigenous people into the system. And if we look at extinctions and colonization together, we see that functional redundancy in native biotas serves as a buffer from functional diversity loss that's caused by extinction. But introduced species are shifting functional diversity in these systems. That might leave some ecosystems vulnerable to a loss in additional functional diversity. And with that, I would like to thank you all for listening. And I'd also like to thank my lab members and collaborators, both past and present, who've made this work possible.
Thank you, Melissa, for that great talk and a great overview of the research being done in the Caribbean with lizards. This is a great time if people have questions that they want to ask Melissa. We already have some that have already been asked, so I will start by putting it also in the chat, Melissa. So the first question is, fantastic talk. With loss in island area, you also see an increase in the number of islands as shelves between the islands are covered by rising sea level. So does this increase in island number have any impact on allopatric speciation that might outpace extinction? And then a follow-up, do islands that were historically connected have similar levels of diversity and functional entities, or are they split up?
Yeah, these are great questions. And I'll start with that second question first. Do islands that were historically connected have similar levels of diversity and functional entities, or are they split up? And we see a bit of both. And I think a lot of that does depend on how the islands were used. And let's see, get this figure. So Antigua and Barbuda, those are two of the islands I pointed out that were previously connected, and they're basically mirror images of one another throughout all these time intervals. They lose a lot of the same species, and a lot of the same species are introduced to those islands. Where we start to see a little bit of divergence, although we see a lot of functional diversity loss in all three of these islands, is in the Guadeloupe archipelago. So that's Grande Terre, La Désirade, and Marie Galante. And Grande Terre and La Désirade are all connected. Marie Galante sits on its own island bank, but it's really close to Grande Terre and La Désirade. And we see the most diversity loss in those three islands, but it certainly is a lot lower native diversity in Marie Galante compared to Grande Terre and La Désirade. I will note that La Désirade is also one of these places that has a weirder history of land use compared to Grande Terre and Marie Galante. La Désirade at one point was a leprosy colony, so you only had a subset of people going there, and it does retain a few of the native fauna that you don't find on Grande Terre and Marie Galante, like Iguana delicatissima, one of the native iguanas. But I think generally, and another thing I guess I'll mention too, is that the Caribbean is interesting because you have these national boundaries that affect biodiversity and how it spreads. So Hispaniola is an island that has two nations on it with different governances and different land use histories. So if you were to subdivide Hispaniola and look at Haiti and the Dominican Republic individually, you'll probably find very different stories about functional diversity. And certainly we've seen that when looking at the mammalian fauna that remains on that island. And that's the case with a lot of these smaller islands as well. So Barbuda and Antigua are both former British colonies. I think they're still British overseas territories. But Anguilla, which I talked about extensively, there are three different European colonizers that have been involved there. You have Anguilla, which is a British overseas territory. You have St. Barth, which is a French overseas territory. And then St. Martin is part French and part Dutch, I believe. So all of those histories, I think, does lead to some levels of divergence in functional diversity. And that first part of the question, let me read that one again. So I'm not too sure about that particular question. You are seeing an increase in the number of islands. I think that's especially the case with the Bahamas. And I'm not sure, I'm not as familiar with the genetic data. I know people are doing a lot of genetic work, particularly with Anolis in the Bahamas in trying to look at whether there has been some incipient speciation, but we are talking about a very short time period here, really only the past 12,000 years. So I don't think there's been enough time for true divergences to occur. But that is a really great question. I think one that definitely requires further inquiry.
Okay, great. Yeah, thank you. Thank you for answering. We have another one that's come through by Sender Sirwan, and he asks, as a response to anthropogenic pressure, have Caribbean lizards undergone a change in lifespans and rate of growth? Any studies on attested growth lines and a change in dietary habitats, isotope studies of the feeding apparatus?
Yeah, those are great questions. And I imagine there certainly has been a change in lifespan just because predation pressures have changed a bit. I know there's been a lot of research in terms of growth rates, particularly with regards to limbs and how limbs, scale with the rest of the body. And this is particularly evident in some of the urban evolution literature. So Kristen Winchell, she has done a lot of research on urban lizards and how their scalation is modified to better utilize urban surfaces that are often sleeker than trees you find in the forest. There's also been some work on how limbs are being impacted by natural selection because of hurricane events. And I'm sure that's something that's been happening over millennia because hurricanes have been a major force in the Caribbean. But as we expect storm activity become more frequent and also more intense, there might be these additional selective regimes that are happening more frequently on lizards. And then in terms of changes in dietary habits, I'm not aware of any successful studies on the diets of these lizards. Again, diet is something that I think we really need to study more and should study more. A lot of these lizards are considered insectivorous, and we're not always clear how much of plant material they're incorporating in their diet. But some recent literature suggests that they do incorporate a lot of plant material in their diet. There's been some studies with the introduced green iguana in Puerto Rico showing that it eats both native fruits and some of these introduced fruits on the island. And certainly if we are changing the vegetation of an island and you have these organisms that are eating vegetation, I imagine their diet's going to change in response to that.
Yeah, thank you for answering that as well. And we have one more anonymous question that's come through. Great talk. I was wondering how you assign ages to the introduction of species during the indigenous and colonial periods.
Yeah, that's a great question. So it was really hard for the indigenous period. And we didn't subdivide the indigenous period as much as we were hoping to because it was really hard to assign dates. There are some specimens that may have been radiocarbon dated, so we have a sense of when it first occurred on an island, or it's associated with particular artifacts that are associated with a particular culture that colonized the Caribbean, because it wasn't just one indigenous wave of colonization in the Caribbean. There were multiple groups that had colonized it at different times and had different material cultures. So that can be helpful, but that is an area that we weren't able to divide as much as we would like. So we kind of group all of these pre-European introductions as one, which is unfortunate, but current data is a limitation. With the colonial period, there actually were, in some cases, written records as to when an organism was introduced. So we utilize those records. And that was also the case with the modern introductions. But as you could see in that figure, one of the figures, there were a lot of taxa for which we do not have an introduction date, most notably in reptiles here. So there were a lot of taxa that we know are not native to the Caribbean, but we weren't sure when they arrived in the Caribbean.
Okay, yeah. We have a few more questions. It's been a great talk. And yeah, so another question is, you mentioned that there are a lot of quaternary fossils in sinkholes. How are you able to sample those, and what kind of time averaging signal do they have?
Yeah, that's a great question. I myself have not sampled those sinkholes. They're a little intimidating, and I haven't personally done any work in the Bahamas myself, which is where most of those sinkholes are. But Those are excavated by cave divers who have, divers who have a cave certification. So that's a really extensive process you have to go to in order to get the certification to go and dive in those caves, because they can be very dangerous to dive into. So I myself haven't worked into them. And in terms of the time averaging signal, I think most of these sinkholes, I could be wrong here, but I think a lot of them are the materials that are coming out of them are Holocene in age. There's been a lot more direct dating of fossils in those sinkholes relative to some of the things you're getting from the caves that I'm more accustomed to excavating.
Okay, yeah, cave diving is like a whole other field. Yeah, completely.
Cool.
We have another question by Sendor, who asks, the introduction of amphibians is comparatively very late compared to mammals and birds. Are there any particular reasons for this? And is the chytrid fungus taking a major toll on the frog population of the Caribbean?
Mm-hmm. Yeah, so I think that's a really great question. I think, one thing I wonder about too with the introduction of amphibians and how late it is relative to some of the other taxa, in this other figure here, we do have some of these that the introduction date is unknown. So it's possible some of those may have been earlier dates. We just don't know. But I think a lot of it might have to do with why people are introducing these animals, with the ones that are intentional. There certainly are a lot of unintentional ones. And during the indigenous and colonial time periods where you have people coming over greater distances, it may have been harder, if a frog got onto a boat, it may have been harder for that boat or the frog to survive the long voyage across the Atlantic, for example, compared to some of these other organisms. But again, I think, my research focuses primarily on reptiles. I am very interested in amphibians, but that's an area we definitely need to do a lot more work with regards to understanding the ancient populations of, and there just hasn't been as much of an investment in that fauna compared to some of these other taxonomic groups. And I'm not as familiar with the chytrid literature in the Caribbean, but I imagine that it is, one of the reasons for decline in these frog populations in the Caribbean as it is elsewhere in the world. It's a great question.
Another question that talks a little bit about ages and aging. You mentioned that not much radiocarbon dating has been done on the fossils from the Caribbean islands. Is that related to tropical ecosystems, which typically do not have much material preserved to allow for accurate radiocarbon ages?
Certainly for the reptile faunas. So collagen preservation is pretty poor, and myself and my colleagues have tried to radiocarbon date some of these reptiles, and they're also very small, and we haven't had much success. There's been a lot more success with dating some of the larger faunas. So, if one finds a sloth bone in a cave and you find a small lizard bone in the cave, there's a lot more to sample from that sloth bone, and it seems that the collagen preservation is a bit better in those larger bones as well. So, and certainly I've tried also to extract collagen for radiocarbon dating from some of the smaller mammals, and we've had difficulties as well. So the preservation is not really great, but there are some sites that have a better context, and people have been able to radiocarbon date them. People have been able to extract ancient DNA from them as well, which is really exciting. And again, I think the size of the specimen sometimes helps with that, because you might have less breakdown of the natural or the molecular components of these specimens if they're larger. But I think also the context in which it's buried is also important there.
Okay, yep. Thank you for answering. One last call for if there are any remaining questions that people would like to ask. One question that we do have that I can pose right now is, with taxon-specific extinctions, are there specific things about the habitats, that is island size or lineage life history, that make groups more or less susceptible to extinction?
Yeah, so I think that habitat affinity, so whether an organism is terrestrial or not, is playing a large role in some of the taxon-specific extinctions we're seeing. And then with island size, I think island size might be playing a role, especially when we start talking about humans on the landscape and humans modifying the landscape. So these smaller islands that have a lot of human impacts on them, you're effectively making the island, a small island, even smaller, whereas those impacts certainly can be drastic on large islands as well. You see on some of the islands in the Great Antilles, which are the largest islands of the Caribbean, significant deforestation after human colonization, particularly European colonization, because you have large-scale sugarcane production. But I think that these effects are particularly acute on small islands. But it's still a little difficult to disentangle the relative roles of these factors because we don't necessarily have really good information on how much historic habitat loss there may have been during the time at which these organisms went extinct. And also, again, going back to the last question with radiocarbon dates, we don't always know when these faunas are going extinct. But certainly with the ones that have been introduced, the ones that went extinct during the time of European colonization, it does appear there is a correlation to the introduction of organisms like the mongoose, the cat, and such, which might mean that particular life history traits might make those organisms susceptible to extinction.
Okay, yeah, great. Thank you again for answering all the questions, Melissa, and thank you once again for a great talk. I will take over the screen share and do a quick outro. Okay, that's the one that I want. Okay, great. Thank you everybody for joining us this week. And we will not be having tea time, but next week, please join us when Ian Castro from the University of Cincinnati will be giving a talk entitled Visibility Critical Risk Theory in Geoscience Field Research, a call to action for new frameworks to assess accessibility and inclusion for field settings. And once again, we have dropped in the weekly feedback form in the chat, as well as the nomination link that you can find now in the chat. And have a great Tuesday, and thank you everybody for joining us, and thank you, Melissa, once again for a great talk and answering all the great questions. Thanks, everyone.
Thank you for having me, Natasha.
Yeah, thank you. Yeah, see you guys next week.